finish that one, and we will start with the adenovirus. All right, so when you're looking at the whole cell infectious cycle for the adenovirus, it just looks a little bit intimidating. So you just need a little bit of time and read the step-by-step the notes, and you will see that it will, after reading a couple of times, it will just make it a little bit easier. So what you are seeing that the adenovirus delivers its genome, which is a double-stranded DNA, and there is a terminal protein attached to the genome into the nucleus. And immediately after that, you can see that the phases of the infection, and we are going to see it throughout, we are going to talk about this a lot. The phases of infection is divided into three distinct parts. You have an immediate early, i.e., early or E, and the L or the late phase. So if I just see that I, E, or L, you know that which phase we are talking about. So the first thing what happens that the transcription happens, the mRNA goes out into the cytoplasm. It makes a specific protein. It makes a couple of different ones, but the most important one is E1A. So this E1A is transported back into the nucleus, and this is the viral transcription factor for the next round or the early protein as well as the later ones. So just remember that the first protein synthesized is the transcription factor for the next one. So that's how most of these schemes will work like that. So now you see the second round mRNAs are synthesized, and they are transported, of course, they are exported out into the cytoplasm, make the protein, and see that this is polymerase, then DNA binding protein, and this is a pre-terminal protein. These are the terminal protein, these are the pre-terminal protein. Altogether, also, the full length, there is different things going on. There is also a protein which is a full length, which is called E2, is formed at this step. The E2 is chopped up in small pieces to make these, but now these are again imported into the nucleus, and see that what they do. They do influence making of some specific RNA that's VA-RNA, virally associated RNA. And this is implicated in some kind of pathogenicity. We are going to see that, some other functions as well. We will see it in the next module. And along with that, you can see that it will also help these proteins will help the expression of the next set of protein, or the L, and also help in the DNA replication, because it's polymerase, DNA binding protein, pre-terminal protein. These are required for the DNA replication. So you can easily see that initial part of the infection, it is just setting up the stage, and then at the last part, these proteins are responsible for the DNA synthesis, and these proteins, see, these are the structural proteins which are made at the very last stage, which together will form the intact viral particle. So you can see that there is a temporal differentiation of immediate, early, and the late phase. The virus do not make all the proteins at the same time, all the RNA at the same time, because that's what is called efficiency. What is needed, it just makes that in a large amount. And that's how the efficiency of these transcriptional programs actually work. It's like it's really mind-boggling to understand that how it is. So the whole thing that what we have seen here, this is summarized in this slide. So connect these two slides, it's very easy in that sense. So the black line, what it denotes, from the very left, this is time 0 towards the viral infection, towards it. So the time starts in there, and it's just an arbitrary. There is no time point or anything that 2 minutes, 3 minutes, nothing. No, this is an arbitrary time point. So, and it is adenovirus and transcriptional regulation. So first one is the immediate early, and see that immediate early, that E1A protein is formed, that we talked about. That influences the early gene transcription right here, influences. In the second round, this E2 is formed. E2 is basically, think about that, is this Pol, DPP, pre-TP, and E2 together. They are formed. You will be able to see that why I'm saying that E2 along with these two names. Now, E2 will, on the other hand, we have just seen that it will basically start the DNA synthesis, because remember that we just saw that polymerase, DNA polymerase, as well as the other factors are also involved in the DNA binding proteins, and the others are involved in that. So once this basically happens, then this E2 is responsible also for the influencing of the DNA replication, and that will basically produce the last stage, these proteins, which is called the 4A2 and the L4. And think about these proteins that they are produced in here, and they influence the late phase, making the structural proteins, which basically will form the entire viral particle. So this is basically the transcriptional program, and think about that this is a little bit different. So when we looked at right here, that LT was just one viral protein. It is doing the viral DNA synthesis by anti-repression, as well as it is influencing the L. And some of these anti-repression molecules were cellular, and just only one protein is involved in here. Here, we have the cascade, first to second, second to third, and so on. So here, the transcriptional regulation is getting more complicated. So that's why we choose these scenarios: one protein, multiple protein, and a cascade regulation. So how basically that E1A, this famous protein, controls the transcription? So what you are seeing that right here, and we are going to see that this basically happens in both cases. Remember that this happens in both cases. This is a part of the DNA. Very carefully understand that. This could be the cellular host DNA, or it could be the viral DNA as well, where there is a binding site for this E2F. E2F is a transcription factor, and DP1 is a dimerization partner. So there is a specific site on the DNA on which these two sits. And on top of this, it recruits another famous protein, which is the RB, or the retinoblastoma protein. So again, remember that E2F D1 recognize some specific DNA sequence, sit on that, and recruit RB. What RB does, RB is a tumor suppressor gene. So the normal function in a cell cycle, is basically, if the cell doesn't need to divide, RB checks it, or it doesn't allow the cell to go on to the synthetic phase from the G1 phase, synthesis phase from the G1 phase. It just arrests the cell cycle. When the cell needs to divide, at the only time that the RB is removed by a phosphorylation, and the transcription goes on to produce the DNA polymerases, which is essential for the DNA synthesis. So that's how it works. So how does this transcription basically works? RB directly don't influence the transcriptional regulation, but it is a little bit indirect function. RB is recruited by this E2F, D2F, and when RB is in there, it recruits these HDACs, which are the histone deacetylases. So now again, remember that we talked about the histone, which makes the nucleosome octamers. And those are responsible for loosening up the DNA or tightening the DNA. You have lots of these basic residues in there, arginine lysines, and this could be acetylated or methylated. When they are acetylated or methylated, then they actually become loose because they don't interact with the DNA and they do not tightly bind. But when you have the histone deacetylases, that means this acetyl group, the methyl groups are taken up. It could be a histone, they take these acetyl group or the CH3 groups out of those residues, the charged residues. And now these amino acids are free. They will interact or attract the the negatively charged the DNA, so it will tightly bind together. So that's the function that the RB recruits the histone deacetylases, which basically takes up these methyl group and the acetyl group from the histones, and that wraps the DNA tightly, and that basically inhibits the transcription. So what E1A does, and we are going to see that many different viral proteins does the same thing. E1A binds to RB, and RB loses the ability to bind to the E2F and DP1. If they cannot bind, there is no recruitment of the histone deacetylases. That means the DNA is loose because there is no methylation deacetylation of the residues. As I said, the DNA is loose and it can do the transcription. RNA polymerase can do the transcription. So that's very important. So this whole thing works in both ways. This works in the cellular host DNA polymerase, in the host cellular DNA, as well as the adenoviral DNA. We are going to see that. Both of them are controlled by this mechanism. So here, on the other hand, we are looking at the adenovirus transcription unit. And again, this is just for your look. Again, you do not have to memorize and say it's like what I'm going to understand from that. Just showing that how complex it is and where the things are, just for your visual thing. There no questions from here. So right here, see that you have the end from 5' to 3' end of the double-stranded DNA with the terminal proteins. E1A, the right here is the E1A. This is the transcription site for the E1A. Then remember that we talked about the second one, E2. So right here, you can see that E2 is this protein, which is chopped up into the polymerase, pre-TP, DNA binding protein. So that's basically what it is. It is the same long one is the E2, and that is cleaved into these different proteins, which will enhance the replication of the DNA and now the production of these proteins, late 1, 2, 3, 4, 5. These are then produced at the later stage. Now here you have the L4 and 4A2. L4 and 4A2 even more enhance the late transcription. So that's just that overview of that, how the transcriptional unit actually works. So fairly complex. We're going to see the herpes virus even more complex, or pox virus even more. All right, so then next we are going to go to the herpes virus, and this is the overview of the cycle. But if you have understood the adenovirus, this is pretty similar. So you can see that it's the infection cycle is divided into immediate, early, late, the same way. But here is a little difference in between the adenovirus and the other one. Remember that for your SV40, if you remember that LT, large T antigen, that was needed. That is needed to be first transcribed. And for adenovirus, we just saw you need then E1A, because that will basically required for the transcription, E1A. These are, if you just remember that these are produced after the initial immediate early gene is transcribed into the mRNA. E1A is produced in the cell. Here, it is different. The first one, so what you are seeing that the herpes virus is getting into the cell, genome is delivered into the nucleus, and it gets circularized, double-stranded genome. It also brings a protein, which is definitely underlined, we will have questions on that, is the VP16. So your LT, E1A, VP16, these are the transcription factors at the immediate early stage for that. VP16 is packaged into the virus. This transcription factor is brought by the virus. It doesn't need to be produced in the cell. So that's the difference between the ones LT and E1A, which is produced after initial transcription. So VP16, basically right here you can see, controls the transcription of the immediate early mRNAs. And these mRNAs produce the proteins which are called the alpha proteins. There are so many of them, you just cannot name them. So it's just a bunch of protein called the alpha proteins. It's just divided into alpha, beta, gamma, just for ease. So the same way, alpha proteins go into the nucleus, influence the transcription of the beta mRNAs, which is exported to form the beta proteins. Now the beta proteins again go back into the nucleus to influence now from the DNA to produce the genomic DNA. What you are seeing that we're going to see that in the DNA replication chapter, here it's not needed. From that double-stranded genome, the herpes virus genome is produced as a concatemer. It's a long molecule. Once the whole genome is produced, it is excised out, packaged into the genome, and it goes on. So it is produced as an end-to-end joined concatemer, that what you are seeing, that this is your genomic RNA, which is packaged into the virus. And at the very last stage, the beta protein also influences the production of the mRNAs, which makes the gamma proteins, and these gamma proteins are the structural proteins. Some of them assemble here inside the nucleus. Some of them are the secretory proteins. These are the goes into the ER and eventually transported onto the membrane of the nucleus, where it basically assembles. So that's the overview of this whole process. So now you can see this is similar to adenovirus, but even more complicated. So the first step, what we are seeing, the VP16 comes packaged into the virus. It is packaged in between. So the top is the envelope, inside is a nucleocapsid, and in between this, there is a tegument protein, and the tegument protein and VP16 is packaged in between the envelope and the capsid. So immediately available after the virus enters into the nucleus. So VP16 influences the immediate early, and at that point, there are two proteins produced. One is ICP4 and another is ICP0. These are the two proteins. So you can see that ICP0 influence early as well as DNA replication, whereas ICP4 enhance the early the early stages, as well as it also influence the late stages. And then you have the replication protein, which are produced as a product of the early expression that influences the DNA replication. So that's the overall mechanism. But what happens that after these initial transcription factors are produced, see, ICP4 stops this immediate early production. So it is a repressor molecule for the immediate early production as well. So what it means that at the initial stage, you produce ICP4 and ICP0. Those are the transcription factor. When they are made, you do not need any more of them. So there is no reason to transcribe those. So it shuts off the immediate early. It switches to early and the late transcription. So this is the precise transcriptional control, so which you can easily see. And you can see that there is a little bit of a blurb in there, but pretty much what I have said, this is all in here on the slide here itself. All right, so that's pretty much of all your transcription and post-transcriptional, and then we are going to look at the viral processing. So any questions before that? You guys good? Okay. So the next part we are looking at, that we are talking about these mRNAs. These mRNAs are produced by the transcription inside the nucleus, most of them. Remember that it's a different case for the RNA viruses. When we're talking about the transcription, this means that these are produced by the RNA polymerase II in the nucleus. These are modified for functioning, and these are what we are talking about in here. The first modification is the 5' cap structure that we keep talking about. 5' cap structure is needed for the ribosome to translate it. Ribosome recognize that. And also the stability of the mRNAs are increased in the cytoplasm for this. And also it will be responsible for the transport into that. So again, ribosome, readability by the ribosome, transport, as well as stability, all of these are required. So how the structure is. So the first enzyme which is involved in that is called a guanylyl transferase. Now, there are many different ways that guanylyl transferase can be there. Cellular guanylyl transferase is a separate enzyme which is a subunit of the RNA polymerase II. RNA polymerase has 12 to 16 subunits. And guanylyl transferase is one of the subunit which makes the RNA polymerase II hollow enzyme, and it happens co-transcriptionally. There are some of the viral guanylyl transferase where the guanylyl transferase activity is also in the RNA polymerase, but it could be a separate enzyme as well. So either it's a part of the RNA polymerase or a subunit of RNA polymerase or a standalone enzyme. Both is possible. But the enzyme function, whatever it is guanylyl transferase. What it does, it brings a G as a cap and of the cap1. This is always a G, a guanosine. And then you can see that right here, this N7 position of the guanine base is methylated. This is done by the guanine 7 methyltransferase. Again, remember that this methylation, this could be a separate enzyme or built in the RNA polymerase. Just depends upon the virus or the cellular environment. In the cellular environment, this is also a part of the RNA polymerase II hollow enzyme. So here you can see a methylated G with the N7 methylation. Now, the second base makes the second and the third base also make the cap2, and this is added to the 5' end of the RNA. The second base is usually, most of the time, is also another guanosine. And here you have a base, but you can see that this base is connected to the first base guanosine, so G and G. Usually that's the normal, there are some exceptions. They are connected by an extremely unusual linkage, and that is what you would take home message. This is a 5' triphosphate linkage which connects these two. So how it is so? So if you just look at the carbon numbers, it is 1, 2, 3, 4, and a 5. And here you are just going the other way. The base is on this side. Here the base is on this side. You're going 1, 2, 3, 4, 5. And what happens is that I'm not going into the detail. Remember that all the nucleotides are triphosphate. It comes with 3 phosphates. So what happens that from this one, so the phosphates are usually connected as alpha, beta, and the gamma. So what happens that this is the alpha phosphate of this. So that's why I see that this is right here in the gray. Whereas here, this is the alpha phosphate, this is the beta phosphate, and there was a gamma phosphate in there. So the gamma phosphate of these of the second base or the cap1 base, cap2 first base or the second base in the whole cap structure, the gamma phosphate of this base is removed, and in here the beta gamma both removed. So this alpha phosphate of the cap1 is connected to the beta phosphate of the cap2. This could be a little bit too much, but that detail is not needed. But just remember that it's a 5 to 5' triphosphate linkage connects these two. That will do it for the quiz or the exam. Then the third base is usually most of the time an A, and an adenine or adenosine. Could be some difference. Sometimes it's guanosine as well, but usually it is GGA is the most common. There are some examples of that. And that is connected to the 3', 5'. You can see just by a regular phosphodiester bond. And then this is connected to the rest of the chain, the same with a 3', 5' phosphodiester bond. This is your regular bonding that is found in the DNA or the RNA no matter what. That's it. So there is another type of methylation happens. Right here you saw the guanine 7 methyltransferase put that N7 methylation. Whereas for the base 1 and 2 of the cap2, this is the second position where you have a hydroxyl. Second position you have a hydroxyl. Here a methyl group is added, and this is done by a 2O methyltransferase. 2, the position, O was an oxygen connected to a CH3. It was a hydroxyl. The H is gone, replaced by a CH3. So 2O methyltransferase. And again, remember that this 2O methyltransferase is a separate enzyme, can be a subunit of the RNA polymerase, or it could be a function of a viral RNA polymerase. All of them could be a part of the RNA polymerase or subunit of the RNA polymerase or standalone enzyme. So all the possibilities we have. So that's your 5' cap structure that is needed. So here in the eukaryotic system or in some of the DNA double-stranded DNA viruses, how it works when the RNA polymerase is basically the enzyme for the transcription. So, as we talked about, that RNA polymerase binds to the promoter and the initiation site, and then even if the transcription factors are not there, the tendency of RNA polymerase is there just to try to transcribe something. In that case, what happens, that the initiation happens, it just goes about 20-30 nucleotides, and if there is no transcription factor, usually the processivity of the enzyme is very, very low. We have seen that TF2H, TF2D, all these altogether comes on to make the RNA polymerase much more processing. If those are not there, just within 20-30 nucleotides, RNA polymerase falls off, it doesn't make a transcript. But if the RNA polymerase goes for 20-30 nucleotides, that means the transcription factors are there. What it does is it phosphorylates the C-terminal domain of the RNA polymerase II. Just again, remember that. RNA polymerase II need to reach at least a 20-30 nucleotide length of making RNA 20-30 nucleotide. Then you have the C-terminal end, CTD, C-terminal end phosphorylation happens. C-terminal domain, CTD. Once this is phosphorylated, this is the site for recruiting of the capping enzyme. This could be guanylyl transferase, methyltransferases, 2-O-methyltransferases, everything. Several different subunits that can be put, those all assembles together. And as transcription is happening, the cap is already made at this point. So this is basically the co-transcriptional capping of the mRNA as it is being transcribed. All right, so after that, so this is your capping. Second part is the modification of mRNA by the polyadenylation or just poly-A tail, basically. So the poly-A tail does have two different functions. One, it increases stability of the RNA, and also it's a nuclear export signal. Without a polyadenylation, those RNAs will not be exported into the cytoplasm, and it increases the stability of the mRNA into the cytoplasm, lives longer. So how this basically happens. And again, if you just look at that, it looks that it is pretty intimidating with so many different proteins, but I'm just going to point out that what are the key steps and what you need to know from here. So the first thing what you have, that you see that the 5' to 3', this is your pre-mRNA. You need to polyadenylate that, and there is a site specifically close to the 3' end, but not at the 3' end. So this is your first important message. We always think that the poly-A tail is added to the 3' end of the RNA. That is true. But it is not at the 3' end of the pre-mRNA. Remember that. There is a recognition site which is called the polyadenylation site somewhere inside, and there is a specific sequence called AAU, AAA, followed by some of these GU-rich sequences. And this site recruits the first protein, which is the CPSF, is the first thing. And then you can see that there are several different proteins, and individual names of these not required at all. So these are all basically the cleavage-specific protein, because you are going to cleave or cut out the RNA from there. CPSF, CSTF, and then you can see cleavage factor 1, 2, 3, 1 and 2. These are all recruited. So you make a multi-protein complex at the polyadenylation site, and that cleaves the polyadenylation site. So now you can see that this part is gone. This was the part of the pre-mRNA. This is gone. So now you have an hydroxyl end open in here. And at this point, you can see that all these factors, they are removed. And slow polyadenylation happens. And the protein that is present in there is poly-A polymerase. So just remember that this is your poly-A polymerase, and this poly-A polymerase, after the cleavage, adds A into this residue at the end in a very slow manner. very slow, about 15, 20. Once it makes a couple of A's, remember that, once it makes a couple of A's, about 15, 20, the last protein is involved, which is a poly-A binding protein 2 or PAB2. Once this poly-A binding protein is recruited onto this, which recognizes the poly-A tail, then immediately the poly-A polymerase gains the processivity, or it just runs into the steroid. It will rapidly synthesize about 200 or 250 poly-A tails, basically. So that's basically the overview of that. So a multi-protein complex recognizes the polyadenylation site, cleaves it, poly-A polymerase is recruited, which does slow polyadenylation. After about 15-20 poly-A's, a poly-A binding protein, which is PAB2, is recruited on the poly-A and result in rapid phosphorylation until you have about 200, 250 poly-A's added to the 3' end of the RNA. That's your polyadenylated mRNA, and that is ready for export into the cytoplasm. So that's basically your take-home message. I will never ask that where CF1, CF2, which step, just the overview of is good enough. So the overview of that, and this is from 2 different chapters. So first, we are looking at the post-transcriptional, that what we are talking about now. In this process, the cleavage of the pre-mRNA is followed by the polyadenylation. Enzyme is a cellular that we talked about. All of these as proteins are CSTF, CPSF, cleavage factor 1, 2, and then poly-A polymerase, poly-A binding protein. These are all of cellular origins. These are exploited by adenovirus, hepatitis B, HDV, herpes, polyoma, retro. All of these use this. On the other hand, there is another different during mRNA synthesis. Remember that. This is after mRNA synthesis. That's why it is post-transcriptional. Pre-mRNAs are done. But these last 2 lines are the first chapter, which is RNA-dependent RNA-directed RNA synthesis. These are done by the viral RNA polymerases, and in influenza and VSV, you have seen reiterative copying of the poly-U residues. Basically, the RNA polymerase slips and added the poly-A's. On the other hand, you can see that for the poliovirus, there is a long stretch of U, which is just complemented by A, that's at the poly-A tail. And in the coronavirus, I just put it as a question mark. It is a much more complicated process in here, and we are going to learn that in the special topic for the coronaviruses. So that's the overview of that when the viral RNA polymerase is synthesized, that is this, and this is right here by the cellular enzymes for the polyadenylation. So the next part we are talking about, that the splicing. And as you can see, let's first take a look at the top left. If you see that right here, you have a green part of the mRNA, then you have a pink part, then you have again a green part. So the green part is written as exon 1 and exon 2. In between them, you have an intron. So in the gene, the coding sequence are not one after another, unlike bacteria. Bacteria, you do not have any introns. They are spaced out with the introns, and these introns are spliced out by the splicing mechanism and joined together. And that actually is the export signal. You need to splice these mRNA before they can go into the cytoplasm. On the other hand, you can see that some viral genomes or the mRNAs can be transported unspliced. This is a special mechanism, so not the normal. You do not see that other than the viral as such. So this was discovered by Phil with the lab of Phil Sharp and Richard Roberts, and this kind of a revolutionary discovery in the molecular biology. So basically, what they found, they were looking at the adenovirus infected cells, and they found these mRNAs are making no sense. Some of these mRNAs, these are called the heterogeneous nuclear mRNAs. Why it is so? Because they were seeing that, okay, all right, these are the proteins. So their mRNAs, if you just run on a gel, should be of this size. But when they're extracting these mRNAs from the nucleus, they are mRNAs. They have caps, they have polyadenylate tails, but they are much longer. And they did not understand that what these mRNAs are actually basically doing. So they did an elegant experiment. So they took the DNA, denatured it so that it is a single-stranded. And they took this hexon mRNA right here, is in the green, and tried to hybridize that, and then they negative stained those and looked under the electron microscope. What they have seen that there are these huge loops of the DNAs which are coming out. And right here, you can see the other part. This is the tangling part of the DNA. And what they saw, this is, of course, this is not the electron microscopic image. This is the colored version of that. And then they saw that this part is the mRNA. And here, although you cannot see, but the end, they saw that this is basically double-stranded. Remember that. What we mean by that, this whole green part right here, this whole green part is basically this part. You have a single-stranded DNA, and the mRNA is hybridized, but you cannot see it in the electron microscope. But at the end, they were two separate pieces, and that made the idea that, okay, all right, this area is hybridized. So I'll just quickly, I should have probably put that picture in there, because that would have been much easier. I change. So right here, this is the picture. So what you are seeing, see that right here, if you're looking at the real electron microscopic picture, this looks like a single strand, but see that the end is bifurcated into two parts. That gave the idea that this is what you are seeing, that this portion is the mRNA right here, which is separated out, mRNA and the single-stranded DNA, and all the black part is the genomic DNA right here. So that's basically what you are seeing in this picture as well. And that gave the idea that there must be these extra sequences which are spliced out, and that just gave the first idea of the splicing. And there was a huge amount of research that started, and it was found that it is a rule in all eukaryotic transcription. Splicing is the rule. So that's basically the process. So here just the loops are being shown, that what is the introns has been shown in here. That's basically what it is, with which the experiment was basically done. So how this basically happens, that's the important part, is when you take mRNA, there are some very specific sites. So let's take a look at that. First, you have an exon 1 in the green. This will be the part of the mRNA. Exon 2, this is in the green, will be part of the mRNA. This intron right here needs to be excised out. And if you look at the sequence, if you just look at that, this is called the 5' splice site. This is towards the 5' end. This is the 3' splice site. So if you look close to the 5' splice site, you can see that these GU, these two residues, are 100% conserved. Frequency of occurrence, 100. This A residue, 100% conserved. And at the 3' splice sites, this AG is 100% conserved. What it means that if you just isolate the mRNAs, hundreds and thousands of mRNAs with all eukaryotic gene, viral gene, and if you just analyze the splice site, those residues, 5' splice site, 3' splice site, and that adenosine, those will be same in everywhere. So that's 100% conserved. And what it basically does. So this splicing is done by a ribozyme RNA catalytic function. RNA, along with protein complexes, hold these RNAs in a proper place and catalyze that. So it's an RNA catalyzed event. So how this happens, that right here you have an exon 1, and the RNA is basically made into a specific shape like that by some of the additional RNAs that we are going to look at the next slide. So what happens that right here, this G, this phosphate, is attacked by the O, which was bonded with this C though. Remember that, but you have the O, but it just does a nucleophilic attack right here, and it causes the first transesterification reaction. So this is excised out. So you have now a 3' OH hydroxyl from your exon 1, and you can see that the 5' splice site of this is bonded by the transesterification with this A. You can easily see, on the other hand, this is the 3' splice site right here, 3' splice site. You have the G. Now this oxygen attacks this phosphate right here, and they are all held in proper space that they have that they can do this catalysis. So this oxygen attacks this, and this is the second transesterification reaction. This basically joins the 5' exon and the 3' exon 1 or exon 2 just by a phosphodiester bond, just a regular 3' 5' bonding. And this is basically the excised lariat, or this loop-like structure. This is basically the overview of that. All right, so that's basically the mechanism. Now here, this slide, and there is a lot of writing at the bottom, and you may be a little bit overwhelmed that what I'm supposed to know that. This is just only for the understanding how these different RNAs, these are the heteronuclear RNAs that U1, U2, you have there is no U3, but U4, 5, and 6. So you have U1 through 6 except the 3. And all these RNAs are in combination with more than 150 proteins. And they make a complex which we call as a spliceosome. And the function of the spliceosome is to bring the sites that we talked about, that GU, A, and the AU, close to each other so that those transesterification reactions can actually happen. This is the 5' splice, this is 5' end, this is the 3' end. So this is your 5' splice site, this is your 3' splice site. So that's what you are seeing right here. You can see that 5', then you have the GU, the RNA is looped all the way right here, and this AG, this is your 3' splice site. And you can see that, again, I do not have to just tell of those, but these are hydrogen bonded. The dotted lines are the hydrogen bonded, and you can easily see that how basically the attacking of these specific residues are happening, and the cutting of the RNAs are done by that. You can easily see right here that this AG, this piece is cut out, and this is the lariat formation. This is the excised RNA. So that's basically the overview of that. And just an overview, in individual names and anything is basically not needed. It is just an overview of that all these heteronuclear RNAs with these spliceosome, which is basically an RNA catalytic ribozyme, place these in a proper position so this splicing reactions basically can happen. That's all you need. All right, so what is the different ways that splicing can be used? In a constitutive splicing that we have seen, say that this is your intron 1, intron 2, these are excised out, and the exons 1, 2, 3 are joined. This is the normal way that it can go. On the other hand, there are some alternative splicing, and we see these more in the viral unit or the transcriptional unit. The reason for that is that this is value added because you have a limited site of the genome, and by alternative transcript from the same sequence, it can make multiple proteins. So that's the advantage of the alternative splicing. So here you can see that exon skipping means you can join 1, 2, 3 or skip the 2 and just join 1 and 3. So these are some of the ones that can happen. An alternative splice site, you can have multiple splice site at the 5' end, or you can have alternative 3' splice site, and all these multiple splice site will make these different combination of these proteins. So basically, from the same sequence of the DNA, you can get multiple different types of mRNA producing different proteins. So here we can look at the alternative polyadenylation as well as splicing for the major late adenovirus transcript. And although it looks very complicated, once I explain that, you will see that it's just take-home message is very simple. So here you have the full the genome. Here is a major late promoter, and that will produce this pre-mRNA. You can see that 5' cap. And right here, these three greens are leader 1, leader 2, leader 3. It's L1, L2, these L's are in small. L1, L2, L3, leader 1, 2, and 3. Then you have capital L1, L2, L3, and 4. These are the different sites. So what happens that this L1, L2, L3, L4, L5, these are all polyadenylation site. So initially, what will happen that you make this mRNA, and after making the mRNA, there is a cleavage right here that, as we have seen, and you have polyadenylation right here. Sometimes the RNA will read through here, and this polyadenylation site can be used, this can be used, this can be used, this can be used. So what we have seen that at the initial stage of the transcription, this first L1 polyadenylation site is used the most. This is the preferential L1 polyadenylation site. So at that time, what happens? Suppose I'm just taking this transcript at the very top to here. I'm taking this one right here. So you can see that written as L1. This is L1, this is L1, I'm just taking. So here what you see that after splicing, these are gone, and these three lines are the tripartite leader sequence that is present in all of them. You have a capped, and then you have a L1, L2, L3. Here you have L1, L2, L3. Then you have the L1, and before that you also have an alternative splice site. That's why you can just get two L1 mRNAs. So you can just see that from this transcript, you can get two of the mRNAs. At the later stage of the alternative splicing, on the other hand, you can see, say I'm just picking up this L3 mRNA. L3 mRNA right here, and it will be the same with the 2, 4, and just two examples are given. If you're taking an L3 mRNA, the 5' end processing is the same way. You're just taking these exons out, so you have the cap, then you have these three lines are your L1, L2, and L3, three leader sequences. These are basically required for translational regulation, for translation. And then you have this L1, L2, and the L3 has an alternative splice site, 3' splice site. So out of those different splicing, you can see that you can get a protein 4, you can get a hexon protein, you get the proteases and other proteins. So you can just easily imagine that just from one transcript, how many different types of proteins you can get. And then you can get this full proteins, L5, L4, you can get L2, you can get those proteins as well, just by regular splicing, not alternative like this. These are extensive for the L1 and the L3. So that's basically the alternative splicing in adenovirus, what you see. Why it does that this bizarre way, that we just really don't have an answer for that. So after this, so polyadenylation is done and your splicing is done, both of them are now required for the nuclear export. That's what we are going to see in the next part. And here also we are going to see that there is a lot of name of the proteins, and not all of them are needed, probably just one or two of these are needed basically. Just the concept is more important. So at the very top we are seeing an unspliced mRNA, pre-mRNA, which is a 5' capped, polyadenylated, and right here these are the heteronuclear RNPC. Basically, this is the spliceosope, nothing but the protein and those RNAs. They are homing onto the intron, and you can see that right here, these SR proteins, these are rich in serine and arginine, and they basically home in towards the 5' splice site and the 3' splice site. Then the splicing happens, and here you can see that a multi-protein complex is formed near the splice site, where the splice site was. That multi-protein complex basically recruit a protein which is NXF1. I did not do the correction. I could have just easily put a structure and put an NXF1, because if you just have the textbook, it is written like NFX1, which is wrong. It's a misprint. NFX1 is a different protein, and that's why I just had to write it down without just making the correction, because it will confuse you. It is in the book, this is wrong. So this is an NXF1, and the NXF1 is, so you will read it as NXF, not NFX, nuclear export factor. So that's very, 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 very easy to remember. So that is the only one that you remember that. So take-home message, after splicing, a lot of protein complexes home in on the splice site. That recruits a nuclear export factor, which is basically, think about that, it's an adapter protein. Once it binds, this helps the nuclear export through the nuclear pore. So the NXF1 can recognize the pore complexes, and it helps the nuclear export. It is an ATP-driven process. It is energy dependent. So that's basically the overview of that. So only one protein, you're just remembering that, which is the nuclear export factor, and transportation happens through the nuclear pore into the cytoplasm. So here on the right-hand side is the same picture. Remember that this is also NXF1, is what we have seen in the eukaryotic system. So this is a cellular pre-mRNA is transported this way. Here what we are seeing that in a simple retrovirus, there is a problem. Remember that. Retrovirus is a plus-strand RNA virus. Plus-strand is basically then mRNA. But the retrovirus is integrated into the genome of the host as a DNA, double-stranded DNA. So that DNA is transcribed into the pre-mRNA. That pre-mRNA will be spliced to make the viral proteins. That is fine. But the problem happens, it has to have 5' end to 3' end, that entire plus strand transported into the cytoplasm to be packaged into the virus. So how this unspliced genome RNA of the retrovirus, which is basically the plus mRNA full length with the introns, is transported, that's what we are going to see here. So here you can see that this is the mechanism by which simple retroviruses, not HIV. HIV is a complex retrovirus. Simple retroviruses, smaller genome, is transported. What they have, very similar. You have this mRNA, and what you have, this is a CTE, which is called a constitutive transport element. This is an extensive secondary structure of the mRNA, which basically recruit the transport factor. Here you can see that nuclear export factor 1 and NXT1 is another adapter protein, and these two protein and also this refs, these are the smaller proteins. So basically think about that, the cellular pre-mRNA, all these are mRNA, all these proteins are homing in onto the splice site. Here on the splice site there is nothing like that happens, but there is a constitutive transport element where the transport proteins home in that help in the transport of that. So this is basically the process. And this your NXF1, NXT2, remember that these are all cellular proteins. Remember that. These are all cellular proteins which are responsible for the transport. So in a small RNA or in a simple retroviruses, the transport is done by cellular proteins. Remember that. That is your take home. Transport is done by the cellular proteins by binding to the CTE. The next one in the HIV, which is a complex retrovirus, the transport is done by a viral protein, not a cellular protein. That's the difference in between two. So what we are looking at here, and again, this part is just for your understanding. It's not needed because retrovirus is a separate chapter. So the top portion is the integrated double-stranded DNA that is integrated into your genome. You have these LTR, those are repeat sequences, and the portion of this, the coding sequence is called a GAG, Pol, and the envelope, and these are proteolized into many different proteins. And what are the different proteins? We're going to talk about that. But what is important right here is just right here, you can look at this, that this is the portion of the REV and the TAT, and right here is the REV and the TAT. In between that, you have this site is needed to be spliced out. This is required for something else, but not for the REV. So we are mainly interested into this protein, REV. TAT and NEF, we not required, but the REV is the most important one in between them. So what happened, you have the full length mRNA after transcription from here, from the DNA, and then what happens that from here, you can see that there is a splicing that happens. And after this splicing, so you can easily see you will have to splice these portions out to join this part of the Rev to that part of the Rev. And this small mRNA is transported into the cytoplasm, which makes this protein Rev. Rev is transported back into the nucleus. And the full-length mRNA, which is a plus genome RNA as well, remember that, the full-length mRNA of the HIV or the retroviruses, complex retroviruses, is a genome RNA as well. There is a secondary structure near the 3' end, which is called an RRE, or the Rev response element. So you have, in the previous one, a constitutive transport element right here, which binds to cellular export protein. Here you have a Rev response element. Rev binds to this and can transport the full length or partially spliced. Remember that it can transport the full length of the genome, or it can transport partially spliced, not where, which makes these different proteins as well. So that's basically, so here the viral protein Rev is responsible for the transport, which can recognize the export, which can be recognized by the nuclear pores to transport that. So that's pretty much all of it.